Hello, my name is Liam Alderson. I'm a junior at the University of Arkansas in Fayetteville, and this is my research project. My project is titled A Cysteine-free Hyperstable Human Fibroblast Growth Factor, Characterization of its Structure and Bioactivity. So my research project focuses on a particular mutant, a cysteine-free mutant of human fibroblast growth factor, which is a protein that is a mitogenic signaling protein in our bodies. It plays crucial roles in multiple physiological processes, particularly embryo development, cell differentiation, cell growth, as well as angiogenesis, which is the formation of blood vessels and tumors. So one of the negative qualities about human fibroblast growth factor, or FGF1, is that it typically has a very low stability whenever it is not bound to its receptor heparin, or whenever it is subjected to high thermal temperatures or low thermal temperatures. The mutation that I'm working with is going to be a cysteine-free mutation. These cysteines contain thiol groups. If you look down here at the structure, cysteine is here on the left. This sulfur is contributing to the thiol group in it. We believe these to be highly reactive, which whenever the FGF gets degraded or it unfolds, these cysteine groups typically form disulfide bonds and negate proper refolding to protein, thus making it useless. We're mutating these cysteine amino acids, which are located here in the structure at points 16, 83, and 117. These all are within the beta barrel motif of the FGF protein. And so we are changing them to serines because these are the most structurally similar. But as you can see, the thiol group has been removed and is replaced with the hydroxide. So we believe this to be structurally similar, but we're getting rid of the thiol groups that we believe makes this protein unstable. The reason that the stability of this protein is so important in the Kumar lab is because there's been evidence and studies done on FGF that show therapeutic potential in regenerative medicine. So this could be used for healing of scar tissue as well as different applications in the clinical world. So as you can see, some of the steps I've already taken, one of the first things we did was purify wild type FGF, because this is what we'll be comparing the mutant to make sure that stability has changed. But as you can see, we ran an SDS page after purifying wild type FGF1 on a heparin cephalose binding column. The wild type FGF, which gets eluted at a concentration of 1500 millimolar NaCl in phosphate buffer solution. And as you can see by this band, which is high intensity, there is quite a bit of the protein that was eluted out of the column. And in lane 8 is our wild type FGF marker. So we know since these bands line up, we did successfully purify our wild type FGF. In the mutant purification, we've started purifying the cysteine-free mutant using glycerol stocks that the Kumar lab already had. And as you can see, first attempts at this purification were unsuccessful. This protein eludes out in the unbound, or the first sample that you load into the heparin cephalose column. And so in both lanes 2 and 3 is where our protein was expected. But as you can see with the gel, it ran and you're not able to distinguish any bands. And so this is something we're going to have to do again to make sure that we're actually getting our correct protein before we can move on to characterization of it. Some of the future plans, we're going to be performing circular dichroism spectroscopy on the protein samples once we have both a wild type and a mutant sample. And this is going to be able to show us any secondary formation changes as a direct result of the mutation. And then we're also going to be testing its thermal denaturation, which will help us understand if there's any greater thermostability within the mutant, as well as trypsin digestion and heparin binding affinity to see if its physiological processes are going to be changed in any way. This research, of course, is ongoing, but it was recently supported by a state undergraduate research fellowship back in January. So this is a project that will be continuing through the year 2021, and I'm very excited to be continuing it. And thank you.